everyone, welcome to my channel Pure Biology. Today I come with another video that is the Golgi complex. Okay, and this is the topic related to cell biology. And before going to this topic, I want to say thank you to all of you who have already subscribed my channel. And this is the, I think, second topic of the cell biology. Before this, I have already uploaded the first topic of the cell biology, that is the lysosome. Okay, so you just check out that video, and after that obviously this one. So look, okay, let's continue the Golgi complex. What is the Golgi complex? This is obviously present both in the plants and the animals. Okay, Golgi complex both present in the plants and animals. And the Golgi complex, endoplasmic reticulum and lysosomes all are three organelles related to each other. Clear? This is generally single membrane bound organelle, single membrane bound organelle, and a part of endomembrane system. Okay? It consists of 5 to 8 flattened membrane bound sacs called the cisternae. Okay, here I have drawn the rough structure of the cisternae. So this is the cis cisternae, this is medial, and this is the trans cisternae. Okay, generally you can say that the one side of the cisternae is the cis position or cis cisternae, and another part is known as the trans cisternae. Suppose this is the cisternae, so you can say this is the cis region and this is the trans region. Clear? So it consists of 5 to 8 flattened cisternae, and each stack of the cisternae is termed as Golgi stack. Okay? This cisternae is called a stack. Okay? Or you can say the dictyosome. The cisternae vary in number of shape and organization. Obviously, here you can see the same structural size or shape of the cisternae, but I have drawn it is very roughly. But generally, the structure of the cisternae is not same, okay, or shape also not same. And about 60 cisternae that combine to make up the Golgi stack in some unicellular flagellates, okay, or you can say that unicellular organism. So there are 60 cisternae attached to each other to form the Golgi complex. In case of mammalian cell, 40 to 100 stacks present. Okay, each Golgi complex that contains 40 to 100 stacks. Here the multiple Golgi stacks are linked together. Okay, this is seen in the mammalian cell. Each Golgi stack has two distinct phases. Okay, a cis phase and a trans phase. So if you consider this is one stack, so its one side is the cis region and its another side is the trans region. Okay. And cis region or cis phase is what? The entry of forming or entry or forming. That means any particle can enter within the Golgi complex through this region, and this is called the cis region. Okay, and this just releases from this region, so this is called the trans region. Clear? Or you can say that here this is the entry path and this is the after maturation the releasing path. Clear? So trans region where the maturation of the particle is completed. So that's why cis phase is called the entry path or forming path, and the trans phase is called the maturing path. Okay? Both cis trans are closely associated with the special compartments, and the cis Golgi network and trans-Golgi network. So here it is saying that the cis cisternae, if it is considered the cis cisternae, this is the trans cisternae and this is the medial cisternae. Okay, so these are present within the Golgi complex. Okay, so that's why here the two cisternaes are present. One is the cis and another is the trans. That's why this is called the Golgi complex, that is containing with cis Golgi network and trans Golgi network. Clear? Cis Golgi network and trans Golgi network. Okay, and the cis Golgi network, the short form I have written here CGN, and the trans Golgi network written here TGN. And the TGN is sometimes called the GERL. What is GERL? G means the Golgi apparatus, E means ER, endoplasmic reticulum, ER, and L is the lysosome. Three are connected with each other. That's why it is called the GERL system. Okay, and proteins and lipids generally enter the CGN, that means the cis Golgi network, and arriving from the ER, endoplasmic reticulum, and exit from the TGN. What is saying that any particles, protein particles or lipid particles that is coming from the endoplasmic reticulum or another part that enter through the CGN. Okay, CGN means cis Golgi network, and this arriving generally from ER, I have told that, and this exit from the TGN or the trans Golgi network. As proteins entering the CGN can either move onwards in Golgi apparatus or be returned to ER. So what is saying here? Any protein particles that is coming from the endoplasmic reticulum that either enter within the Golgi apparatus through CGN network, and either it enters within the Golgi apparatus or be returned to the endoplasmic reticulum. Okay, not remain any part of the cell. Either it enters within the Golgi apparatus or it return back to the endoplasmic reticulum. Similarly, any proteins which exiting from the TGN can either move onwards and be sorted or be returned to earlier compartment. So if maturation is complete, sometimes maturation is complete of the protein, because protein particles enter the Golgi apparatus for maturation. Okay, so after maturation, it either that is coming out through the TGN network or trans Golgi network, or it remains within the Golgi apparatus. Clear? And after coming from the TGN of the Golgi apparatus, then it is sorted. Okay. Golgi apparatus are prominent in secretory cells. Okay. Generally, the cells which are secretory, that means they secrete any substances, there the Golgi apparatus is needy organelle, okay, or important organelle, you can say. Example is that goblet cell of intestinal epithelium which secretes polysaccharides. This is example. Another cell you can take obviously as example. So I just give the example of goblet cell. Okay. This cell of the intestinal epithelium is very important. Why? And here the Golgi apparatus present very importantly, or you can say the many Golgi apparatus are there. Okay, so this secretes the polysaccharide rich mucus into the gut. So here Golgi apparatus must be present, because without presence of Golgi apparatus no secretion is possible. Okay, so that's why it remains there. Then Golgi apparatus is often considered the distribution and the shipping department for the cell's chemical products, obviously, because generally Golgi apparatus help in releasing some secretory products. So that's why these are called the distribution and the shipping department of the cell. Clear? And it modifies the protein and the lipids that have been built in ER. So what is the function of ER? ER generally produce the protein or lipid particles that are necessary for the cell. But the protein or lipid particles that are produced in the endoplasmic reticulum, there the maturation is or processing is not possible. So just for processing they have to go in the Golgi apparatus. Clear? And after completion, if this is important for the cell, so that means that remain. But if this is not important for cell, that means these are digested by the lysosome. Substances from the ER enter into the C phase. That's why ER and the C phase of the Golgi apparatus, these are closely located. Clear? And the and the trans phase is found near the plasma membrane. Why? Because after processing through the trans phase, the proteins and the lipid particles just comes out. Okay. So that's why this trans phase of the Golgi apparatus that remain near the plasma membrane. Okay. So this is actually the structure and the function of the Golgi apparatus I have just explained here. So it is completed here. Okay. So I think you all understood this. And about Golgi apparatus, I will try to discuss another topic, obviously in my second part of this video. Clear? So thank you.